Alright, good morning, Dr. So. This is our Medchem presentation. Firstly, I will talk about the flower that I chose, which is Iris domestica, also known as leopard lily. It has a long history of use in traditional Chinese medicine. It is a bitter cooling herb that acts mainly on the lungs and the liver, lowering fever and reducing inflammation. It is effective against a number of bacterial, fungal, and viral organisms, and has also been used as an antidote to snake bites. Some of the active ingredients in the roots are flavonoids and isoflavonoids. Some examples of flavonoids are tecdorin and iridine. And some examples of isoflavonoids is beta-carotene. Flavonoids are an antioxidant, anti-inflammatory, anti-cancer, antimicrobial, antiviral, anti-angiogenic, anti-malarial, neuroprotective, anti-tumor, and anti-proliferative. Flavonoids can inhibit phosphodiesterases such as CAMP, phosphodiesterase. CAMP is a second messenger molecule key in the regulation of different cell functions during inflammation. High levels of CAMP have been associated with anti-inflammatory functions. Therefore, flavonoids can decrease inflammatory by inhibiting CAMP. Flavonoids are also potent antioxidants able to scavenge free radicals and to decrease their formations. The roots of leopard lily are used especially in treatment of inflammations of the throat and upper respiratory tract, including acute laryngitis, acute tonsillitis, edema of the glottis, asthma, and cough with profuse sputum. Juice of the roots is used in Nepal to treat liver complaints, where it has the benefit of improving the appetite. This juice is also used to abort a fetus during the first trimester of pregnancy. Now I'll be passing the time to Quan Chen, who will be talking about his plant.
Okay, so I'll be talking about Pakya, speciosa, also known as petai. Okay, next. So it is widely found in Southeast Asia, such as Malaysia and Singapore, and it is used in local cuisines and traditional medicine for various ailments, and it has anti-diabetic, anti-inflammatory, and antioxidant properties. Um, next, looking at the research that I found, it shows that in Singapore, it is the seeds of the petai is eaten raw to treat diabetes. So I will be talking about how um the mechanism behind it. So it contains flavonoids such as quercetin, m- myricetin, catechin that acts as a competitive inhibitor on alpha glucosidase and alpha amylase. These are enzymes responsible for carbohydrate digestion in the intestine. So by inhibiting these enzymes, there will be a decrease in glucose availability for absorption, thereby lowering postprandial blood glucose levels. Okay, next. So these are the this shows how the flavonoids inhibit the enzymes by looking at its structure. It has this hydroxyl group found on the beta ring right here, which forms hydrogen bond with the catalytic site of the alpha amylase. Thereby, the starch cannot bind to these enzymes. Therefore, the glucose cannot be formed. So next, uh, the flavonoids also modulate voltage-gated calcium ion channels, enabling the calcium ions to enter the cell to activate the calcium-dependent protein kinase 2, which helps to facilitate the fusion of the insulin containing vesicles uh, with the plasma membrane, such that the insulin will be secreted out of the cell, promoting exocytosis, and it affects the increased uptake of glucose by cells, thereby decreasing the blood glucose levels. Yeah, I will pass it on to Emily.
Okay, so now I'll be talking about my plant, which is Flamingo strobilifera, which is also known as wild hops. So it belongs to the Fabaceae family, which is also known as legumes. It is commonly used in traditional medicine and it has analgesic, anti-inflammatory, anti-convulsive, antimicrobial, anti-ulcerogenic, and anthelmintic properties. The bracts, leaves, flowers, and roots of the plants can be found to be useful in folkloric medicine. So some of the uses, the roots can be used in the treatment of indigestion, fevers, epilepsy, and hysteria in Bangladesh traditional medicine. It can induce a heavy sleep and relieve pain. With a sufficient dose, heavy sleep can be even induced under severe pain. The root juices can also be used for diarrhea and dysentery. The leaves, a decoction or infusion of leaves and flowers can be used in the treatment of tuberculosis and as a vermifuge for children. Arabians use it in cosmetics as anthelmintic and as a remedy for coughs and cold. The roots are rich in different secondary metabolite groups, which could be responsible for the plant-derived medicinal properties. So studies have shown the presence of terpenoids, and methanolic extract was found to be a source of natural antioxidants to prevent progress of various oxidative stresses. Flavonoids, flavonoid glycosides, chalcones, epoxychromines, and pterocarpans are the main constituents found in Stropilifera species. So a few examples of the active ingredient can be found in the root extracts are flavanone, genistein, beta-sitosterol D-glucoside. So I'll be briefly explaining it. So flavonoids are bactericidal, bacteriostatic. It damages the cytoplasmic membrane and inhibits energy metabolism and synthesis of nucleic acids. Genistein, it is a phytoestrogenic isoflavone with antioxidant properties, and it plays a role as an antineoplastic agent, a tyrosine kinase inhibitor. Beta-sitosterol D-glucoside acts as an inhibitor of cholesterol influences that trigger inflammation. Now, I'll be giving you an example of the product that can be made from Flamingia stropilifera. So one example would be Nature's Green Flamingia root granules. It is used as a product in traditional Chinese medicine, and its function is that it expels winds and damp to strengthen the waist and knees. So now I'll be passing the time on to Evo.
Okay, thank you, Emily. So now I'll be talking about my plant, the Balaria lupulina, more commonly known as the hop-headed Balaria. So this plant belongs to the Acanthaceae family, and it is commonly used in folklore medicine of Southeast Asia, India, and South China. The hot aqueous extract of Balaria lupulina can be used to treat insect and snake bites, inflammation, allergic responses, as well as diabetic-related complications. So here are some of the active ingredients present in Balaria lupulina. These are 4-methyl catechol, 4-vinyl catechol, and 4-ethyl catechol. So when it comes to treating inflammation, these active ingredients activate the nuclear factor erythroid 2 pathway. This neutralizes the reactive oxygen species by reducing the amounts of free radicals present. This will allow us to increase the cortical arrangement of actin, reducing stress fibers. There is also an increase in cell-to-cell adhesion due to the increase in claudin 5 to form tight endothelial junctions, strengthening the endothelial barrier function. This will overall lead to a decrease in microvascular leak, allowing for decreased inflammation. So when it comes to treating diabetic complications, it also comes about through the activation of the NRF2 pathway. When the NRF2 pathway is activated, this binds to the heme oxygenase 1 gene, allowing for the production of the HO1 enzyme. The HO1 enzyme enhances antioxidant, anti-inflammatory, and anti-apoptosis capacity of the tissues. This will allow us to reduce the degree of damage in diabetic complications by protecting the vessels. So now I'll be passing my time over to Ryan to talk about his plant.
Okay. Thank you, Evo. Now I'll be talking about my plant, Labicia pumila, otherwise known as Kachip Fatima. Now, Labicia pumila is a plant that's native to Indonesia, Malaysia, and Indochina, and its roots and leaves are widely used to regulate menstrual cycles and expedite childbirth. In the extract of the leaves and roots, there are multiple active ingredients. However, a few of them are gallic acid, pyrogalol, camphorol, and myricetin. However, the main active ingredient in the extracts is gallic acid, as it has high bioavailability as compared to the rest. Now, the uses for gallic acid in Labicia pumila extract is mainly menstruation by helping to relieve menstrual cramps and to treat pelvic inflammatory disease by helping to reduce the negative effects such as inflammation, bleeding, and uterine damage. And how gallic acid does this? Is through its anti-inflammatory effect, and it achieves its effect through the regulation of the NF-kappa B pathway, which is involved in inflammatory and immunity response. Now, one section of this response pathway that is very crucial to its effect is the section that's circled in red, whereby the P65 protein has to first undergo acetylation by the P300/CBP dependent histone acetyltransferase in order for its complex to undergo dissociation. However, gallic acid can act as a non-competitive inhibitor of the P300 CBP dependent histone acetyltransferase, thereby preventing P65 from undergoing acetylation and thus preventing its complex from dissociating. Without the dissociation of the complex, it cannot enter the nucleus and transcribe the gene that is required in order for the inflammatory and immunity response to occur, and as such, there is less inflammation that will occur, resulting in less painful menstrual cramps as well as less immunity response, so there's less damage to the uterine lining in the case of pelvic inflammatory disease. Thank you.